Okay, while the stragglers are coming in, I'll refer to the announcement board. I will have office hours today from 9 to 10, and it seems maybe we have a room down the end of the hall around the corner from the GSI office that might be available again. Very important reviews for the midterm. It gets very quiet. There's one on Sunday afternoon in 2060 VLSB run by several of the GSIs. And there is my review Monday night, 2050 VLSB, run by me, I guess. And both of those are questions and answers. And a question that is not acceptable is, can you explain respiration to me? That's a whole lecture, okay? So that's not an acceptable question. The exam handout, very important, is on B space. What's on the exam handout is where you have to go for your midterm on a week from today on Friday. So please check that out. Today I'm going to talk about photosynthesis, but I want to make one comment concerning respiration. There's a figure 117 that I didn't refer to at the end of last lecture, and I'll just take 1 minute to point out what's in there. What figure 117 shows you is that glycolysis and the Krebs cycle are the source of a lot of compounds that are used for the biosynthetic reactions, processes. If you look at the bottom of the Krebs cycle, you see, for example, succinyl coenzyme A. Succinyl coenzyme A is a precursor, the starting material for the synthesis of heme, something we talked about a lot last time, and also chlorophyll, something that we'll talk about a lot today. And there are various other points in these pathways, like phosphoglyceric acid is a precursor for serine, things like this. So there are really two things going on when you are running the respiratory pathway. You are, of course, making ATP, but you're also producing starting materials for the synthesis of amino acids, nucleotides, prosthetic groups, etc. So this is something you should be aware of. Okay, today we're going to talk about photosynthesis, and I am almost exclusively going to talk about photosynthesis in higher plants. The reaction is carbon dioxide plus water producing sugar plus oxygen. The reversal of respiration. Therefore, the free energy change for this reaction is plus 686 kilocalories. The energy that is required comes from H nu, which is light. Initially, I want to mention something because it's relevant to the lab next week. I want to just mention the Hill reaction. The Hill reaction is a reaction discovered by very unusual man. All these English scientists of the 20th century were unusual. Robin Hill. And the question he was dealing with was, where does this oxygen come from? Does it come from CO2 or does it come from water? Pretty simple, pretty easy question. So what he did was he ground up some material, leaves, probably spinach, like you're going to be doing, and he measured oxygen evolution in the light using not the normal electron acceptor that occurs naturally, but some artificial compounds like you're going to use. You're going to measure the Hill reaction with a compound known as DCPIP or DPIP. Other compounds will work, but he measured oxygen evolution in the absence of carbon dioxide. There was no carbon dioxide in his reaction medium, and this was the first demonstration that oxygen came from water. So that's why the Hill reaction is a very important result in the history of photosynthesis. Subsequently, when people obtained isotopes of oxygen, radioactive isotopes or heavy isotopes, they repeated this experiment and showed conclusively that the oxygen comes from water. So what's happening in this reaction is carbon dioxide is being reduced to carbohydrate, and oxygen is coming from water. Water is being oxidized to produce molecular oxygen. a very unusual reaction, which we will talk about more. The reaction I have on the board here is called oxygenic photosynthesis because it produces oxygen. It occurs in both eukaryotes and prokaryotes. The prokaryotic organisms that carry out oxygenic photosynthesis are cyanobacteria that you looked at when you had your microscopes out a week ago. There are organisms that are known as carry out an anoxygenic photosynthesis where there is no O2 production. You need an electron donor to reduce CO2 to carbohydrate. They do not use water as that donor. They use strange compounds such as hydrogen sulfide, which is converted to sulfur in their reactions. This reaction only occurs in prokaryotes. I won't talk about it, but you should be aware you have oxygenic and anoxygenic photosynthesis. This is a scheme that is similar to the one in figure, lost my book already, 118. It shows the light reactions and the so-called Calvin cycle or CO2 fixing reactions occur in different processes. These processes can be separated. For example, the light reaction box, here's light, produces oxygen from water. It also produces products. These products should look somewhat familiar to you. ATP is a product of light reactions. And NADPH, which is the brother of NADH. There's a phosphate group on the ribose ring. That's the only difference. Otherwise, you can, if you understand NAD, NADH, you should understand NADP and NADPH. This is an electron carrier. This is the energetic compound produced. The products of the light reactions are used in a series of other reactions, which used to be called the dark reactions. That is a terrible misnomer because these reactions are occurring during the day, not in the dark. So we now call them the carbon fixation reactions, the carbon fixing reactions. In Berkeley, we like to honor our distinguished scientists of the past, so we call them the Calvin cycle. Calvin's lab was basically next door to this building. So Calvin was an organic chemistry professor. So I'll talk about that next lecture. And in the second series of reactions, carbon dioxide using ATP and NADPH is converted to sugar, okay? The reactions of photosynthesis in eukaryotic organisms, not cyanobacteria, occur in a special organelle, just the way the reactions of respiration, not all of them, but many of them occur in a special organelle. And that, of course, is the chloroplast. There are lots of pictures of chloroplasts in figure 123. They're a little hard to see, but you can, I think, get the idea. I draw a chloroplast like this. There's an outer membrane, and then there is this complicated internal membrane system. You have, the membranes take two forms. They stack up. It's like a series of pennies or coins. You have a stack of membranes, and these stacks are connected by unstacked membrane regions. The names are important. The stacks are called thylakoids, or they're called grana. The unstacked membranes are just called stromal membranes. The region around the membranes, there's a liquid region, which is known as the stroma. Now, coming back to this, well, it's on this board. The light reactions of photosynthesis, which are producing ATP and NADPH, you should be able to see why you would expect those reactions to occur in the membranes. And fortunately for all of us, much of what I say today is a reiteration of what I've said about respiration, because electron transport in chloroplasts has a lot of similarities to electron transport and ATP synthesis in mitochondria. So we've got the membrane fraction, which is carrying out this box. It's the portion of the reactions that are interacting with light. And then we have the Calvin cycle or CO2 fixing processes which are occurring in the soluble stroma phase of the chloroplast. I just drew this to point out that when you have these stacks, this is a stack, the membrane encloses a space. There's a space in here, and it takes the same name as the internal space that was used for the rough ER. It's called a lumen. It's not called a matrix. Matrix is only used for the internal space of the mitochondrion. So you have a luminal space inside the stacks. Now, light. We have to talk a little bit about light. Sunlight. Sunlight is a form of energy, electromagnetic radiation. In classical terms, light was characterized by wavelengths. Different wavelengths of light have different energy contents. In quantum mechanical terms, light was also then characterized by containing packets of energy. The packets of energy are known as photons or quanta. These two terms are used interchangeably, and we still talk about the wavelengths of light, but we also talk about quanta and photons when we talk about light. The energy of a photon or a quantum of light is inversely proportional to the wavelength. That means UV light, 200 to 300 nanometers long, is very energetic. It's so energetic that it can break chemical bonds, and this is why you should not sunbathe. Because you sit in UV light, you're going to start breaking bonds, and you're going to get bad things happening, let's say, put it that way. Visible light in the 400 to 700 nanometer range is less energetic and can be detected. These are the colors of wavelengths of light that we can see. Infrared light, further than, say, 700 nanometers, is of even lower energy. Visible light is what's used by plants in photosynthesis. The range of wavelengths are 400 to 700 nanometers. That wavelength of light, I'm going to start to erase things because I don't need this. 400 to 700 nanometers. The energy is approximately 40 to 50 kilocalories per quantum, or per photon. Okay? So you can see there's a lot of energy in a quantum of light. Remember, 686. Well, the nice thing about light is it's free, relatively speaking, and cheap, and it is very energetic. So there's a lot of light, there's a lot of energy available to carry out that synthetic process, the overall reaction of photosynthesis. However, for light to be used, it has to be absorbed. A lot of light is coming down onto this planet from the sun, and is not absorbed, it's just never used. Molecules that absorb light are called pigments. And when light absorbs light, several things can happen. When light, did I say light? When a pigment absorbs light, several things can happen. The first thing that happens is the light is absorbed and you produce what is known as an excited state. So this molecule now has the energy of one quantum of light energy, and it's excited. You know, it goes, ah. It's like when you have too many cups of coffee in the morning. And this is what's shown in figure 119. The photon is being absorbed. The pigment is converted into an excited state. Now, several things can happen to this excited state. Some of them are good, and one of them is good. Some of them are bad. One thing that can happen is the excited state can decay to the ground state and simply give off heat. Nothing happens. You warm up. Secondly, you can actually transfer the energy from this excited state to another molecule with very little loss of energy. This is physics. This is not biology I'm talking about. And that actually happens in photosynthesis. I'll talk about that in a minute. The light can be re-emitted. If the light is re-emitted, that's called fluorescence. In general, this is not a good process for us, us being plants. But what is a good process for plants is something that's called charge separation, where the excited state pigment forms a positive and a negative compound. There is what's called a charge separation, or it's also called a photooxidation, where an electron is ejected out of the pigment, leaving a plus charge, and that electron goes to someplace, a molecule in the membrane, for example, and is known as an electron acceptor. So what you're doing in this situation is you're converting light energy into chemical products. Very important. Once we have chemical products, a cell knows how to deal with chemical products. I mean, when I talked about respiration, all of those things were either, particularly in the electron transport chain, those were oxidized and reduced compounds. And here we have an oxidized compound and a reduced compound. So we have gone from physics to biochemistry, essentially, by carrying out a charge separation act with the pigment. What are the pigments in photosynthesis? The main pigment, and I'm looking now at figures 120 and 121 and 122. I'm not going to draw any of this on the board because it's too complicated. But the main, everybody knows the main pigment in photosynthesis is chlorophyll. The structure of chlorophyll is shown in figure 120. If you look closely at that molecule, and I hope you can recognize that it looks a lot like heme. It's got these 4 nitrogen group rings that are surrounding not an iron atom, but a magnesium atom. So that is a major difference between chlorophyll and heme, is that there's a magnesium in a chlorophyll and an iron in a heme. But another substantial difference is chlorophyll has a long, very, very long hydrophobic hydrocarbon chain. What does this mean? It means you would not expect chlorophyll to be floating around in solution out here, but you would expect it to be found in the hydrophobic region of the chloroplast, which are these thylakoid membranes. In plants, chlorophyll comes in 2 forms. There's chlorophyll A and there's chlorophyll B. What do I want to erase? Nothing. So chlorophyll A, chlorophyll B. These differ slightly in structure. There's a C2H5 group in chlorophyll A. No, there's a methyl group on one of the rings in chlorophyll A and an aldehyde on one of the rings, the same ring in chlorophyll B. So chlorophyll B is a little more polar than chlorophyll A, and you're going to work with those in lab next week, and that will be important for how you separate these things out. There are other pigments that are found in photosynthetic organisms. For example, plants. There are carotenoids. The carotenoids generally are hiding because there's so much chlorophyll, you don't see the carotenoids until the chlorophyll starts to break down in the fall, and then you get red leaves and you get yellow leaves, and they're really pretty. And when I teach this in the fall, I can bring in these wonderful colored leaves and show you, but there aren't any now. But carotenoids you're aware of as beta-carotene, for example, which is found in carrots, and other carotenoids as well. And then there are some pigments that I don't really have to refer to in any detail that are found in cyanobacteria. Cyanobacteria have this blue-green color because they have pigments which are known as bilin pigments. Not terribly important. Each one of these pigments has a distinct absorption spectrum, and the most interesting ones are the chlorophyll. So this is wavelength, and this is absorption. You're going to do this next week. And chlorophylls are characterized by 2 absorption bands, one in the 400 nanometer range, and one in the, let's say, 650 to 700 nanometer range. That's what's shown better in figure 121. Figure number 3, the trace number 3, and that is chlorophyll A, and the trace number 2, I guess, it's hard to tell, is chlorophyll B. Chlorophyll A and B have different absorption spectra because they are chemically different. The carotenoids are figure 4 in that they absorb light in this range out here from, say, 500 to 600 nanometers. Why are there different pigments in photosynthetic organisms? The important point here is that you want to absorb as much light as you can. If it isn't absorbed, it isn't used. So by having these multiple pigments, you're extending the region of absorption over the visible region. If you didn't have the carotenoids, there would be a region here of very little absorption by chlorophyll. And once this energy is absorbed by these pigments, I mentioned it's possible to transfer it. So you can transfer energy from carotenoids to chlorophylls, from chlorophyll B to chlorophyll A, things like that. So it just has to be absorbed. I'll make this point now, and then I'll come back to it again. Chlorophyll is always found in association with proteins in photosynthetic complexes, photosynthetic organisms. So we have chlorophyll protein complexes. That is, the chlorophyll is not simply free in the lipid bilayer floating around. It's attached to proteins. Proteins are obviously very hydrophobic proteins. They're integral proteins buried in the membrane, and they bind many chlorophyll molecules. It's not simply one-to-one. It can be 10 chlorophylls per protein or more even. If you look at Table 122, there's an important message there. It shows the distribution of pigments in oxygen-evolving organisms. The important point is that the only pigment that is found in all oxygen-evolving organisms is chlorophyll A. Chlorophyll B is found in some. For example, it's found in plants. It's found in green algae. But chlorophyll A is absent from cyanobacteria. I should point out that cyanobacteria do a photosynthesis that is almost identical to what happens in higher plants. So there's no distinction there. I mean, obviously it's not exactly the same, but I'm saying almost the same. But they can do that photosynthesis without chlorophyll B. Chlorophyll B is obviously a non-essential pigment. An aside, this is an aside. There is an organism known as Halobacterium halobium that I like to mention at this point. Halobacterium halobium is a salt-loving bacterium. It's not a true bacteria. It is now in the Archaea class, which are unusual bacteria-like organisms. And it grows in, I don't know, 4 molar salt or something. The interesting feature of this organism is when it grows anaerobically. It can grow aerobically or anaerobically. When it grows anaerobically, it makes a pigment, purple pigment. The pigment is called bacteriorhodopsin. And I actually talked about the structure of this when I talked about membrane proteins. This is a pigment that contains a rhodopsin molecule. A rhodopsin is the pigment we have in our eye. It's a very primitive organism, primitive pigment, and it's got a light-sensing pigment in its membrane. You can see this organism if you ever fly over San Francisco Bay. You're coming into San Francisco Airport. Not so good from Oakland, but San Francisco. You fly over these salt marshy lands, and the salt concentration is very high. And if you look out, those, you'll see this purplish color, reddish purplish color. That's Halobacterium halobium, and you can turn to your seatmate and impress them enormously by telling them that's Halobacterium halobium. And do you know what it does? It does photosynthesis. What this organism can do is it can absorb light. This pigment will absorb light, and somehow that induces a cis-trans conformational change in the pigment, and that allows a proton to move from one side of the membrane to the other. A proton gradient is established, and guess what? It can make ATP. So that's photosynthesis. Without chlorophyll, no chlorophyll. This is the only organism known that does not have a chlorophyll-based photosynthesis. And if this doesn't impress your seatmate, you can spill your coffee on them or something. The spectra of these pigments. Why is chlorophyll green? Because it doesn't absorb. This is the blue spectral region, and this is the red spectral region, and it doesn't absorb the green light. So chlorophyll, the green light is passed through, and we see green from chlorophyll. The carotenoids are yellow because they're absorbing pretty much in that region, greenish region or yellowish region, but they are also, they absorb some of this, so they are yellow and orange and red. Remember, the presence of multiple pigments allows for greater absorption of light energy. No energy, no absorption, nothing is going to happen. Pigment function. Pigments do two things. They function. One is antenna. And antenna means just that. You've got a big, like, antenna which absorbs light, takes in light energy. And if you look at figure 124, it shows you there is an array of the dark molecules are antenna pigments. There's a lot of antenna pigments. Light is absorbed by those antenna pigments. And so this is simply absorption. And transfer of energy. So antenna pigments are dealing with energy. Antenna pigments. And ultimately, what happens to this absorbed energy is it is transferred to a special site in the chlorophyll protein complex, which is, you can see in the middle, which is known as the reaction center. And at the reaction center, charge separation occurs. This is the site where the absorbed light energy is converted into chemical products. So you can see that you're not going to have any photosynthesis if you don't have reaction centers. Potentially, you might have photosynthesis if you have no antenna. Why? Because the antenna aren't carrying out the charge separation. Photosynthesis, you can make mutants that have almost no antenna, and they work, but they're not happy, healthy organisms. Let's put it that way. So what happens at the reaction center? There is, firstly, the reaction center is a pigment protein complex. Containing a very small number of chlorophylls. Small number of chlorophylls. And these are all chlorophyll A molecules. This is why chlorophyll A is absolutely essential for photosynthesis. So when I say a small number, it's usually 4 to 6 chlorophylls in reaction centers in plants. Small number of chlorophyll molecules which are involved in what I call charge separation. And what happens is this. There's a special form of chlorophyll in the reaction center. Chlorophyll A, chlorophyll A. It is a dimer. Two chlorophylls like a sandwich laying over each other. This was proposed on the basis of sort of spectral measurements for years and years and years. Finally, we now do have, I took them out this year. I didn't want to drive you guys crazy. We do have the structure, X-ray diffraction structures of reaction centers from plants, and indeed, there are two chlorophylls that are just lying close together. This dimer in the light gets converted to an excited state, like that, and then it loses an electron. So we end up with a dimer now that has a positive charge and an electron which has been transferred to some acceptor A minus. This reaction, as written on the board, is known as the primary reaction of photosynthesis. It's the conversion of light energy into chemical products. It's a very unusual reaction. It is what makes everything on this planet possible. It turns out that in plants we have not one but two reaction centers. And they go by simple names: reaction center 1, reaction center 2. I like that. Yeah. What is the H nu? Is there an H nu somewhere? Dimer, this says dimer. Chlorophyll, this is the chlorophyll dimer. That's light. That is light. That is light. Okay, I could do this if you're happier. I can do that. H nu, that's light. Okay, converting the dimer to an excited state, and then the excited state converted to a charge separated state. 426. 4-6. There are approximately, vary slightly in different reaction centers. My point here is that there are very few chlorophylls in the reaction center. That's what's shown in that figure. But there's a lot of chlorophylls in the antenna. Okay? Okay, so I was saying that there are two reaction centers in plants, 1 and 2. They have different special chlorophylls. So this one has a chlorophyll known as P700, which undergoes oxidation in the light. This one has a chlorophyll P680, which undergoes oxidation in the light. These are different. These reaction center complexes contain different chlorophylls. They contain different acceptors, and they contain different proteins. So they're not identical in any regard. You'll notice that the wavelength at which these two absorb are slightly different. This one absorbs more effectively in what is known as far red light, longer wavelength, lower energy, and this one in red light. When you have a reaction center it is associated with a large number of antenna chlorophylls. For example, each of these reaction centers have associated with it approximately 200 chlorophyll A molecules, chlorophyll B molecules, and carotenoids. That's true for photosystem 1, and it's true for photosystem 2 as well, other pigments. This combination of antenna plus a reaction center is what we call a photosystem. So now we've gone from a separated group of antenna. We've included that, associated that antenna with a reaction center. So we get a photosystem, which is reaction center plus antenna. And again, the large number of antenna that are found in a photosystem are to facilitate the absorption of as much light as possible. And that light energy is transferred to the reaction center. The reaction center is where the action occurs. This reaction that I've written on the board is a very unusual reaction. It's not, it's physics. It's solid state physics. It is very, very fast. For example, the estimation is this reaction occurs in 10 to the minus 12 seconds, which is 1 picosecond. It is independent of temperature. You can take membranes and cool them down to liquid helium temperatures, 1 degree Kelvin, okay? This reaction will still occur because there's no movement. It's all hardwired in. There's no movement of molecules or atoms that are occurring in this process. You've got everything sort of lined up structurally so that you give it a flash of light, and in 1 picosecond, you form the products. So that is an amazing feature of this process. You have to be clear about the difference between these two photosystems. When I say photosystem 1 and photosystem 2, what are the differences between them? And I'll reiterate that. They have different set of chlorophyll binding proteins. That's why they absorb light in different regions. Although they both have chlorophyll A and chlorophyll B, they have different proteins. And they have different, what are called reaction center chlorophylls, which is very important. Okay, what happens after this light is absorbed and we get a charge separation? Well, what happens is there is an interaction between the two photosystems. And what's shown in figure 126A is what we have. Here is photosystem 2, and here is photosystem 1. And when light is absorbed by this, you get an acceptor reduced and a oxidized chlorophyll. When light is absorbed by this, you get an oxidized chlorophyll and acceptor reduced. This electron is used to re-reduce this acceptor. It needs an electron, right? It'll go down there. This electron up here is going to go to NAD to form NADPH as a product. And the ultimate source of electrons in this system is water. Water is being oxidized to produce oxygen, 4 hydrogens, and 4 electrons. So the electrons are re-reducing the P here, and then that electron is passing through and ending up in NADPH. In NADP to form NADPH. That's probably more accurate. This is what's happening. NADP plus is being converted to NADPH. The source of electrons for the reduction of NADPH is water. Water oxidation, releasing electrons. What's produced in the light in photosystem 2 is a very strong oxidizing agent, the strongest oxidizing agent known in a biological system. This compound, P+, P680 it is, P680+, I'll be more specific here. P680 plus formed in the light is capable of oxidizing water, and this, as I mentioned before, this is the only place in biological systems where water is oxidized. You can imagine if we had a lot of water oxidation going on, it wouldn't be very good for our planet, wouldn't be very good for us. In the other photosystem, this oxidant is not as strong, P700 plus, but this is a very, very strong reducing agent capable of carrying out this particular reaction. Now, what happens is this electron transfer from A, I'll call it A2, how's that, to P700 is not direct. It's not as I've drawn it here. What happens, and here's where things start to look like mitochondria. We have the A2 acceptor, which has an electron in it. Okay? That electron is transferred to a compound known as plastoquinone, PQ. PQ looks very similar to ubiquinone in mitochondria. So that's kind of interesting, but important. What happens to ubiquinone in mitochondria? Ubiquinol when it's formed. Anybody want to tell me? What complex does it react with? 1, 2, 3, or 4? 2, wrong. 3, right. Well, it turns out that chloroplasts and all oxygen-evolving organisms have what is known as a cytochrome BF, which is equivalent to a cytochrome BC complex. There is a complex that is structurally similar to complex 3. It is functionally similar to complex 3 in that it oxidizes plastoquinol. The electrons go through this cytochrome complex, and then there's a difference. Where do the electrons go from complex 3? Remember? Cytochrome C. Well, in plants, there is a beautiful, lovely protein, blue protein, known as plastocyanin, that accepts these electrons. Pretty blue color. Beautiful. So there's a copper protein here. It's blue, and it contains copper. There is no cytochrome C. This is a peripheral protein, just the way cytochrome C is a peripheral protein. The electrons pass through plastocyanin, and they go to photosystem 1, to P700. So you've got an electron transport chain that is transferring electrons from the acceptor of photosystem 2 through a cytochrome complex, through a copper-containing protein, to the reaction center of photosystem 1. What do you get out of this? You get oxygen. Way back here, I'll make it more complete. P680, this is photosystem 1. Photosystem 2, I'm sorry. What you're getting as products so far is oxygen, okay? And up here, the electrons are going to the A1 acceptor to form NADPH. So you're getting oxygen as a product and NADPH as a product. This series of reactions, as drawn on the board, is known as the non-cyclic electron transport chain. It's non-cyclic because the electrons start in water and end up in NADPH. Now you might say, is there a cyclic electron transport chain? And the answer is yes, and I'll talk about that. I'm not sure when, maybe not today. What's the other product of the light reactions other than NADPH? ATP, great, thank you. Where do you think ATP is made? How do you think ATP is made? It's made by a chemiosmotic process. Look closely at figure 126b. What that shows is the complex is in the membrane. The membrane is sort of runs through this thing. And you can see that as these electrons are transferred through this non-cyclic pathway, firstly, you get protons over here when you oxidize water. These are not moved across the membrane. They're in the luminal space because water oxidation occurs in the luminal space. But when this plastoquinone and plastoquinol are formed and oxidized, there is an additional movement of protons across the membrane into the lumen. So what we're doing in chloroplasts is we're increasing the acidity of the lumen, not the outer surface, the outer volume, which is the stroma. Protons are being moved from the stroma across the membrane to the lumen. And no surprises here. If you look to the right of figure 126, you see there is an ATP synthase sitting in the membrane. Its orientation is opposite of the orientation in mitochondria. That is, the protons move through the ATP synthase, and ATP is synthesized in the stroma. NADPH is synthesized in the stroma. Remember, the Calvin cycle enzymes are in the stroma, so you have no necessity to move things across a membrane. You synthesize both NADPH and ATP in the stroma. I'll mention one compound because it's going to be, you're going to use it in the lab. There is an inhibitor of this process, a classic photosynthesis inhibitor that blocks right here, known as DCMU. DCMU prevents the reduction of plastoquinone. It's sort of the classic non-cyclic electron transport inhibitor. So when you add DCMU, are you going to make ATP? No. Are you going to make NADPH? No, because none of the electrons that get up to A2 can get out of A2. And you will see, I think, for yourself in the lab next week how this works. I want to say one thing, last thing, and then I'm going to have to finish up on Monday with this. One experiment that I'd like to mention at this point, I told you about the problems that Mitchell had having people accept his results. Well, a lot of the experiments that provided the best evidence for the Mitchell scheme came from photosynthesis. Why? Because you don't start, the reaction can sit there and nothing happens. There's no real substrates until you turn the light on. Then all kinds of things happen. For example, you can measure pH changes very conveniently with chloroplasts. But there is an experiment known as the acid-base experiment that is just so clever that it has to be talked about. You have chloroplast membranes, and you put them at pH 4. And the lumen, you let them sit there, and the lumen will become pH 4, pH 4, pH 4. Okay? Then, very suddenly, you make the external pH 8. So now you have 4.0 inside, 4.0 inside, 4.0 inside, 8 outside. Okay? You add ADP and phosphate. And guess what happens? You make ATP. Why do you make ATP? Because you have artificially created a pH gradient across the membrane. This is done in the dark, does not require light. It's done in the presence of DCMU. There's no electron transport. There's no light reactions. You have simply artificially created a pH gradient across the membrane, and that gradient can be used to synthesize ATP. This, I think, was the death knell to the people who kept looking for chemical intermediates. There's really no way to explain this unless you take the chemiosmotic mechanism very seriously. So this is a really great experiment. I wished I had done an experiment like that. Okay, on Monday, I'll talk a bit more about this and then we'll talk about carbon fixation.
Like, how is the A minus? Is the A minus a primary reaction receptor?
Yeah.
Yes.
Yeah.
Oh, okay.
You don't have to know what it is.